Hello, my name is Museum Wadia and today I'll be presenting my task 2, that is prediction using unsupervised ML, and basically we'll be discovering the iris dataset to predict the optimum number of clusters and represent it visually. So first of all, as we all know that we have to import all the required libraries such as pandas, numpy, matplotlib, seaborn, sklearn. So after that we will read the data that is given to us in the CSV format, that is pd.readcsv, and the name of the file. And let's explore the data that we just imported. So we can see there are around 1, 2, 3, 4, 5 columns, and the ID column is not usually that useful to us, but there's data about the sepal length, sepal width, petal length and petal width, most probably the structure of the plant that we are talking about. So now we'll look at the different species that we are talking about. Okay, so it's iris setosa, iris viscolor, and virginica. And if I could show you what we are talking about, is something like this. So this is what we are talking about, and the petal size and the sepal size are the different structure of this plant, and we are talking about the size of the petals, as you can see here, and the sepals, as you can see over here. Now let's check how many null values that we have in the data set. So data2.isNum.sum, we get the sum of all the number of null values that we hold, and we are lucky enough not to have any null values that can distort our learning or our end result. Now let's look at the head. This is the same thing, just the top 5 rows of the data set. We have a look. Okay, so now I'll quickly tell you the basic difference in unsupervised learning or machine learning. The data structure or the data that is provided to us doesn't have any label. It has data that is, we can evaluate or visualize, but it doesn't have data that is specifically labeled. And through clustering, that is what we're going to do, we will get to know the different sets of data that are closely related to each other, and later on the sklearn cluster import k-means will help us differentiate different kinds of data into groups. So it will create a group of data that is similar to each other and put aside the data that is not similar. And we also know that k-means is the way of unsupervised learning through which we can actually use the elbow method, which can help us understand or get the value of k, that is the value of clusters that we need to define in order to understand the data and the sets of data that will be created later on. So now plot the result on the line graph, allowing us to observe the elbow. Elbow is basically the inflection point. The graph will show you an inflection point, and that inflection point will be the value of k, which will be the most optimized and the most best value that we can use in order to find the number of clusters. So let's go ahead, and here we see that using the basic syntax, this, and plotting the graph by the elbow method, we see the number of clusters and the inflection point. We can say the inflection point could be this point, and this is the value at around 3. So let's try to keep the value of k as 3. Now as you see that we're gonna apply the k-means to the data set, creating the k-means classifier. Now we understood that how we found the value n clusters equal to 3, because the inflection point lies around here. And using the fit predict method, Further, we are able to create a model through which we are going to plot a scatter plot so that we will get the clusters of the data set. So now I will use the scatter plot method, and we will label each scatter plot with the species that we are given, that is all the iris species. And further, plotting the centroid of the clusters. Centroids are basically colored as yellow, and also above every cluster is given a different color in order for us to visualize it in a better way. So now after plotting, we get to see that almost every species in a different color. That is, iris setosa is in red color, iris versicolor is in green, blue, and iris virginica is in green, and all the central points are plotted, which is actually looking quite appropriate if you go to see. And it successfully collects the data points that are similar to the species and differentiate between the data points. So we have successfully completed this task.